Assalamualaikum, welcome back to my video. We're still discussing on lesson learning outcome, which is number 4, light dependent reaction. So let's discuss on the second types of electron flow in light dependent reaction, which is the cyclic photophosphorylation. This is the schematic diagram of cyclic photophosphorylation. What you see here is that only photosystem 1 involved in cyclic photophosphorylation and only ATP produced in cyclic photophosphorylation. There is no production of NAD, pH, there is no involvement of photosystem 2 in cyclic photophosphorylation. So cyclic photophosphorylation generates surplus ATP that satisfying the demand of Calvin cycle in order to produce the food. And some organisms, for example purple sulfur bacteria, use this photophosphorylation to produce ATP and to produce food. And cyclic electron flow is thought to have evolved before linear electron flow. Linear electron flow referring to the non-cyclic photophosphorylation. So let's see the process involved in cyclic photophosphorylation. So again, in the middle of this diagram is the thylakoid membrane. The inner part is the thylakoid space and then the outer part is the stroma. So photosystem 1 embedded in the membrane of thylakoid, and there is series of protein complexes involved in electron transport chain of cyclic photophosphorylation. It is the protein ferredoxin, and then there is a cytochrome complex, and then there is a plastocyanine. So let's discuss how ATP being produced in cyclic photophosphorylation. So photon hits a pigment, and then the energy from photon is transferred from one pigment to another until it excites the reaction center of photosystem 1 P700. In P700 there is electron, so energy is being transferred to the electron, causing electron to be excited to the primary electron receptor. Excited electron from P700 is transferred to the primary electron acceptor, causing P700 to oxidize, forming P700 plus. But there is no photolysis involved here because eventually the electrons that is missing from P700 will be replaced by the electron itself. Alright, so what happened to the electron in primary electron receptor? Let's see what happened here. So the excited electron will force down an electron transport chain in a circle from the primary electron acceptor of photosystem 1 to protein ferredoxin, and then to cytochrome complex, and then next to plastocyanine, and eventually to the reaction center complex of photosystem 1 P700, from which it was ejected. So during the falling down of electron in electron transport chain, there is release of energy. So energy released by the falling down of electron drive the creation of proton concentration gradient across the thylakoid membrane that later leads to the diffusion of hydrogen ion across the membrane and drive the ATP synthesis using chemiosmosis in the next video that we will discuss later. So there is huge difference between cyclic photophosphorylation and non-cyclic photophosphorylation. What are they? Number 1, photosystem involved in non-cyclic or photophosphorylation both, which is photosystem 2 and photosystem 1, while only photosystem 1 involved in cyclic photophosphorylation. There is photolysis process happen in non-cyclic photophosphorylation, and no photolysis occur in cyclic photophosphorylation. Since there is photolysis in non-cyclic photophosphorylation, so there is release of oxygen in non-cyclic photophosphorylation and no release of oxygen in cyclic photophosphorylation. And then the flow of electron in non-cyclic photophosphorylation is in linear, whereby the electron move from photosystem 2 to the photosystem 1, while in cyclic photophosphorylation, the electron flow in a circle from photosystem 1 and end up in the photosystem 1 again. And then the final electron receptor of non-cyclic photophosphorylation is the electron carrier, the NADP+, and the final electron receptor of cyclic photophosphorylation is the photosystem 1 itself. And then the products in non-cyclic photophosphorylation is NADPH and ATP, while in cyclic photophosphorylation, the product is only ATP.